I'm I meant to have you write it at the top of your notebooks at the first day of the semester, and I totally forgot. Your AP exam, if you want to turn to the first page of your notebook so it's big and bold, or just wherever, your AP exam is May 14th. May 14th. So we'll have the exam. It's a Friday. Um, yes, is during school day. So it is a Friday, and then we have uh, one more full week after that, and then 3 days in that next week. So exam, full week, and then a couple 3 days. So we'll probably spend that last week doing lab stuff, just like doing basic like skills. Yes. Goodness gracious, no, I'm not giving you a final. Um, if you're working hard towards one exam, I'm not gonna give you another one a week later. If I have to give you a grade, like for report card purposes, it'll probably be overlappy stuff, maybe like a quiz over the techniques we learned, but nothing major. I wouldn't do that to you. So yeah, May 14th, AP Bio exam, then one more full week, and then a Monday, Tuesday, Wednesday, and the last day of school is that Wednesday. Okay. Any other questions? Today is all about meiosis and genetic variation, which is like a big deal for college board people, which is why I had to switch gears and actually make this a notey thing instead of more explorative. So we're going to start out with a summation comparing meiosis and mitosis. So we're going to do a little chart. So this is meiosis and genetic variation. Okay, and we're going to start off with mitosis versus meiosis. So we're going to do a chart. The first column is going to be the property we are discussing. So like what characteristic? I'm going to put mitosis first because we learned mitosis first. And meiosis. So we're going to start with ploidy. So, oh, it's sort of shaky. It looks like it's shaking. Okay, so meiosis can only happen with diploid cells. So you can't divide a haploid cell in half. That's the whole point of meiosis, is to cut the number of chromosomes in half. We can't have a half set of chromosomes. So meiosis can only happen with diploid organisms, where meiosis can happen in diploid or haploid. Is it shaking? Okay, I don't like that at all. Okay, maybe it stopped. Okay. Mitosis can happen with diploid or haploid organisms. So we talked about the life cycles where we had full-blown like multicellular haploid organisms. Those haploid organisms need to grow. In order to grow, they have to undergo mitosis. So you can duplicate a cell if it is diploid or haploid, but you can't undergo meiosis unless you are diploid. So there's that. DNA replication. It's another big one. DNA replication in mitosis happens in interphase. Before before mitosis happens. In meiosis, it also happens in interphase, but only before meiosis 1. There is no DNA replication between meiosis 1 and meiosis 2. It only happens once. It happens before meiosis 1. Crossing over is another really big differentiation point. Also a really big portion of what today is all about. It does not happen in mitosis. Crossing over does not happen in mitosis. That's a big ol' X. Or apparently a fish. Big ol' X. In meiosis, it happens in prophase 1. Not prophase 2, just prophase 1, when duplicated homologs pair up. So, crossing over is a big source of genetic variation and a big thing we are going to talk about. It happens during prophase 1. It does not happen during prophase 2. All right. Okay. The next one is the number of daughter cells. In mitosis, we get 2 genetically identical daughter cells. Unless something dramatically horrible happens. In meiosis, we get, these are also, never mind. And in meiosis, we get 4 haploid and genetically unique daughter cells. So if a haploid cell goes under mitosis, don't know where to emphasize. If a haploid cell undergoes mitosis, you get haploid daughter cells. If a diploid cell goes under mitosis, you get diploid daughter cells. They are going to be genetically identical. A diploid cell goes into meiosis, you get 4 haploid cells out of it. And our last little column is the role. Why on earth do these 2 separate practices exist? So, in mitosis, we are producing cells for growth. Repair, or in the case of asexual reproduction, or in the case of asexual reproduction, reproduction. Doesn't sound grammatically correct, but it is. So, it's really, really messy. I'm sorry. So mitosis is all about growth, repair, all that fun stuff. And if something like a bacteria cell reproduces, mitosis is all for that as well. Meiosis produces gametes or spores. And reduces chromosomes by half. So that is our generalized overview of mitosis versus meiosis. Any comments, questions, concerns, screams of anguish? We're going to move on to the good stuff. So, all about genetic variation. Genetic variation. So, genetic variation is produced in sexual life cycles and contributes to evolution. So, genetic variation produced in sexual life cycles and contributes to evolution. Your exit ticket for today is a little bit lengthier, mostly because it's asking you to draw the connections between genetic variation, the things we're going to talk about, and evolution. So let's see. Yes. So in sexually reproducing organisms, most of the genetic information comes from chromosome behavior in meiosis. Most of the genetic variation comes from chromosome behavior in meiosis. And there are 3 mechanisms. Oh, that's really messy. 3 mechanisms for genetic variation that we are going to discuss. So, 3 mechanisms. The first is called independent assortment. Independent assortment. Okay. The idea behind independent assortment is that during metaphase 1, right, when all of those duplicated homologs come together, they meet along the metaphase plate, they are randomly oriented, meaning so all the chromosomes in your cells right now, right? You have one set of chromosomes from your biological mother and one set of chromosomes from your biological father. We call those the maternal and paternal chromosomes. So every cell in your body has maternal DNA and paternal DNA. So when you meet up at the metaphase plate, in some places, mom's DNA is going to be on top and dad's DNA is going to be on the bottom, and some dad's going to be on the top and mom's going to be on the bottom. And that is just how they're oriented how they're oriented or how they're sorted determines which gametes get maternal chromosomes and which gametes get paternal chromosomes. So to put that in words, during metaphase 1, the homologous pairs each consisting of one maternal chromosome and one paternal chromosome are randomly oriented along the metaphase plate. So our gametes, when we were, so in the case of biological female humans, all of the eggs that you have were produced when you were in utero. So, not only does that mean that at one point half of your genetic information was in your grandmother's uterus, it also means that we've, like, this is all done and accomplished. But I mean, some of your gametes have more of your biological mother's DNA, and some of your gametes have more of your biological father's DNA, just because of random assortment of this stuff, which is why sometimes you look a lot more like your biological mother's side of the family, and sometimes you look a lot more like your biological father's side of the family. For example, I have a little sister. She's 2 years younger than me. I look a heck of a lot like my mom's side of the family, and she looks a lot more like my dad's side of the family. So if we were to draw this out, let's say we have 2 cells. They both have a big chromosome or they have a big maternal chromosome and a small maternal chromosome, and they have the homologous pair of paternal DNA. If they're oriented like that along a metaphase plate, but they could also be oriented like that. If they undergo meiosis 1, this cell is going to get all of the maternal DNA, and this cell is going to get all of the paternal DNA. This cell is going to get the small chromosome from maternal DNA and the large chromosome from paternal DNA. This cell is going to get the small paternal chromosome, and this is going to get the large maternal chromosome. If we undergo meiosis 2, We undergo meiosis 2. We're going to get 4. Oh no, what happened here? Go back. This chromosome's going to get 1 long and 1 short chromatid. Same here. Same here. This one is going to get the long paternal and the small maternal. This one's going to get the opposite. This means for an organism with 2 chromosomes, right? For an organism with 2 chromosomes, there are 4 possible combinations. This is combo 1. This is combo 2. This is combo 3. And this is combo 4. So in the production of gametes, an organism with 2 chromosomes, or an n equals 2, there are 4 different possible gametes for them to make. Now, when it comes to humans, right, we have a lot more than 2 chromosomes. So the number of combinations, the number of possible outcomes increases dramatically. In fact, we have a little math equation. The number of possible combinations of chromosomes when assorting independently is 2 to the n. So the number of possible chromosome combinations when assorting independently. Goodness gracious, I need to slow down. Is 2 to the n. So in humans, n equals 23. So the number of possible combinations of maternal and paternal chromosomes of maternal and paternal chromosomes is 2 to the 23rd, or 8.4 million. So there is a 1 in 8.4 millionth chance that you got the combination of chromosomes from your maternal grandmother and maternal grandfather that you did. So there's a 1 in 8.4 millionth chance that you got the DNA from your biological mother that you did, or 1 in 8.4 millionth chance that you got the DNA that you did from your father, or biological father. Mm-hmm. We'll talk a little bit more about that when we get into genetics. It all really depends. So if we're just looking at one chromosome or one gene on one chromosome, then the math gets a lot simpler. So if your maternal grandmother has a gene, then there's a 50-50 chance your mom got it, and then there would be a 50-50 chance that you would get it. So like a 1/4 chance that that specific trait passed down. But also the traits that you got from your father can interfere, cancel out, or strengthen those traits. That's the first way we get genetic variation in humans. The second way we have talked about already, and that is crossing over. There we go. Yes. Okay. Crossing over produces something called recombinant chromosomes. Recombinant chromosomes. A recombinant chromosome is any chromosome that carries genes from two different parents. So, in the creation of your mother's gametes or your father's gametes, their parents' chromosomes all line up, and there's a chance that a little bit of grandma's DNA mixed with a little bit of grandpa's DNA, and you got chunks of both. So that's why genetics a lot of times isn't that simple. In humans, there's an average of 1 to 3 crossing over events per chromosome. In humans, there's an average of 1 to 3 crossing over events per chromosome. This means that the orientation of sister chromatids also further increases the genetic types of daughter cells. So if we draw it out, actually I'll do it on here so you have it later. If we've got maternal and paternal, right, and they do a little crisscrossy, and then this one goes this way, and this one goes into 2 other cells. It can be arranged like this, or like this. So depending on which way that the crossing over sister chromatid happens, could increase the variation in final gametes. So sister chromatid orientation also increases variation. And this right here, this orientation is in metaphase 2 because we're worried about sister chromatids. So we're in meiosis 2. Does that make sense? Okay. So that's our second form of genetic variation, or second source of genetic variation. The last one is random fertilization. So, it is completely random which egg gets fertilized by which sperm at any point in time. It's not like there's a specific order that eggs are ovulated. So, each gamete has a 1 in 8.4 million chance of getting a certain chromosome combination. So, where'd it go? Each gamete where are we at? Yes. Represents a 1 in 8.4 million chromosome combination. So the random fertilization of a male gamete with a female gamete is 1 in 8.4 times 1 in 8.4. So random fertilization produces a zygote with any of 1 in 70 trillion diploid combination. This means with any two parents, there is a 1 in 70 trillionth chance that they got the combination of DNA that you did. So you are more than 1 in a million. You are 1 in 70 trillion. And this is without taking crossing over into account. Woo. So when you think about the possible genetic combinations, it's a heck of a lot more than 1 in 70 trillion. But we can only do the math for that. And that's not even accounting for the fact of the probability that any two individuals do wind up reproducing. This is just within one pair of human beings. So it would essentially take more than 1 in 70 trillion kids to get two that look exactly alike from two separate fertilization events. Yes. So identical twins happens after fertilization. So when an egg is fertilized, right? So fertilization event happens. We have a zygote. That zygote at any point in time has the likelihood, like if a meiosis event, if mitosis goes too far, can actually split in half and create two zygotes, which is how we get identical twins. So identical twins happen after fertilization. Fraternal twins is when two different fertilization events happen. Good question. Any others? No. Okay. So this is very, very important for evolution because genetic variation is the reason why we get more combinations and more genes appearing in a population. So keep evolution in mind as you fill out your exit ticket. It's a little bit heftier, so you might have to save it and come back to it. So I'm not sure if Canvas saves your text responses if you exit out. So to be safe, if you don't think you're going to get it done, I do it in a Google Doc and copy paste. So if you have any questions, please let me know. We didn't talk about evolution, so I'm asking you to do the critical thinking.